In this video, we'll explore the key concept of transport in plants, which allows them to grow as high as 100 meters. These are topics of the standard level and higher level IB biology syllabus. We will review transpiration, the structural adaptations of xylem vessels, the distribution of tissues in a dicot stem and root. And for higher content, we will go over how root pressure contributes to water movement and how the phloem is adapted for translocation. Finally, we'll review how sugars move through the phloem in a process called translocation. Water movement through a plant begins in the roots, where root hairs absorb water from the soil by osmosis. This occurs because the root hair cells have a lower water potential due to the active uptake of mineral ions. Once inside the root, water travels through the cortex towards the vascular tissue in the center. From there, water enters the xylem vessels and begins moving upwards through the stem. This upward movement is primarily driven by transpiration pull, though root pressure and capillary action also assist. When the water reaches the leaves, it moves into the mesophyll cells and evaporates into the air spaces within the leaf. The water vapor then exits through tiny openings on the leaf surface called stomata. This loss of water through the stomata generates a negative pressure in the xylem, effectively pulling more water upwards from the roots in a continuous process known as the transpiration stream. Gradients play a crucial role in transpiration by driving the movement of water from the soil through the plant and into the atmosphere. The key type of gradient involved is the water potential gradient, which is the difference in water potential between two areas. Water always moves from areas of high water potential, with more water and less solute, to areas of lower water potential, with less water and more solute. In a plant, the water potential is highest in the soil, lower in the root cells, even lower in the xylem and leaf cells, and lowest in the air outside the leaf, especially on a dry, windy day. Let's take a closer look at how transpiration is the result of water evaporation from the plant into the atmosphere, ultimately playing a large role in the humidity of ecosystems. In plants, the evaporation of water plays a vital role in water transport, but also temperature regulation. The evaporation occurs primarily at the leaves, though small amounts of evaporation may occur in stems or flowers. Inside the leaf, water is delivered by the xylem, part of the plant's vascular tissue. From the xylem, water moves into the spongy mesophyll cells, which are loosely packed with large air spaces that facilitate gas exchange. As water reaches these cells, evaporation happens when liquid water from the moist cell walls of the spongy mesophyll cells turns into water vapor. These cells are located inside the leaf and are loosely packed with air spaces around them. The water vapor then moves towards the stomata, surrounded by guard cells, which are small pores mostly found on the underside of the leaf. When the stomata are open, water vapor moves out of the leaf into the surrounding air due to a water potential gradient. Inside the leaf, the air spaces near the spongy mesophyll cells are moist and have a high water potential because of constant evaporation from the cell walls. In contrast, the external air, especially on dry, warm, or windy days, has a much lower water potential. Because water always moves from an area of higher to lower water potential, water vapor diffuses out through the stomata into the drier atmosphere. This loss of water vapor is a passive process driven by this gradient, and it plays a key role in maintaining the transpiration stream, which helps pull water up from the roots through the xylem. This evaporation and diffusion of water from the leaf creates a transpiration pull, generating negative pressure in the xylem that draws more water upwards from the root. When water evaporates from the moist cell walls in the leaf and exits through the stomata as water vapor, it creates a negative pressure or suction inside the leaf's air spaces. This loss of water needs to be replaced, so water is pulled from the xylem in the leaf veins. That, in turn, pulls more water upwards from the xylem in the stems and roots. This continuous upwards flow is possible because water molecules stick together, a property called cohesion, and also stick to the walls of the xylem vessels, a property known as adhesion. These forces help form a continuous unbroken column of water all the way from the roots to the leaves. The movement of liquid through narrow spaces without the help of external forces like gravity is called capillary action. In plants, the forces help water rise through the xylem vessels. Transpiration pull moves water upwards through the plant, especially during the daytime when stomata are open and transpiration is high. Capillary action and transpiration pull work together to move water upwards through a plant's xylem. Xylem vessels are highly specialized structures in plants that are perfectly adapted for efficient water and mineral transport from the roots to the leaves. The cells form long hollow tubes made from a series of dead cells called vessel elements, which have no end walls. One of their key adaptations is the walls of the xylem are thickened with a substance called lignin, which provides structural strength and prevents the vessels from collapsing under the tension created during transpiration pull. Lignin can be deposited in different patterns, such as rings or spirals, allowing flexibility while still reinforcing the vessel. The narrow column and lignified walls creates a continuous column through which water can flow freely without interruption. Another important feature is the presence of pits, small, unthickened areas in the walls that allow water to move laterally between adjacent xylem vessels or into surrounding cells, helping distribute water through the plant tissues. Additionally, xylem cells have no cytoplasm or organelles such as a nucleus, which reduces resistance and allows water to move quickly and smoothly through the vessels. Their narrow diameter also plays a role as it enhances capillary action, helping water to rise upwards even in the absence of root pressure. These adaptations, working together with physical forces like cohesion, adhesion, and transpiration pull, make xylem vessels extremely effective in transporting water and dissolved minerals to all parts of the plant. Let's look at a plan diagram of a dicotyledonous or dicot plant stem. This is a simplified representation showing the basic structure and distribution of tissues, not the fine details of individual cells. In this transverse section of a dicot stem, several distinct tissue layers can be observed. The epidermis forms the outermost layer, serving as a protective barrier and sometimes containing a waxy cuticle to reduce water loss. Beneath the epidermis lies the cortex, composed mainly of parenchyma cells that store food and may carry out photosynthesis. Separating the cortex from the central tissues is the cambium, a thin layer of meristematic tissues found between the xylem and phloem in the vascular bundles of dicot stems. Its main function is to produce new xylem cells on the inside and new phloem cells on the outside, contributing to the plant's secondary growth, that is, the increase in thickness or girth of the stem over time. Inside, the vascular bundles are arranged in a ring, a defining feature of dicot stems. Each bundle consists of xylem, which is oriented inward for transporting water and minerals, and phloem, which is oriented outwards for transporting sugars. At the core of the stem is the pith or medulla, made of parenchyma cells that store nutrients and water while also helping to support the stem structure. Now we turn to the transverse cross-section of a dicot root, shown here as a plan diagram. Like the stem, the epidermis forms the outermost layer, but in the root it includes root hairs, elongated extensions that greatly increase surface area for water and mineral absorption from the soil. Just beneath the epidermis lies the cortex, a broad region made mostly of parenchyma cells, which store starch and allow water to move toward the center. The inner boundary of the cortex is marked by the endodermis, a single layer of tightly packed cells that contains the Casparian strip, a waterproof barrier that forces water and solutes to pass through the cells allowing the plant to control what enters the vascular system. At the center of the root is the stele or vascular cylinder, which houses the transport tissue. In a dicot root, the xylem is typically arranged in a star or cross-shaped pattern at the very center. Between the arms of this xylem star lie patches of phloem, responsible for transporting sugars and organic compounds. In some cases, especially in older roots, a ring of cambium may develop between the xylem and phloem. This is a layer of meristematic tissue that contributes to secondary growth by producing new xylem and phloem, similar to the role it plays in the dicot stem. Overall, while the stem and roots share some tissue types, their organization differs to suit their function. The stem supports upward transport and structure, while the root focuses on absorption and entry regulation of water and nutrients. Let's take a closer look at the root hairs extending from the epidermis tissue. Root hairs are specialized extensions of epidermal cells that play a crucial role in water uptake by significantly increasing the surface area of the root in contact with the soil. These thin, finger-like projections extend into the surrounding soil, maximizing access to water and dissolved minerals. Water enters root hair cells by osmosis, moving from the higher water potential in the soil to the lower water potential inside the root hair cell. Plants actively transport mineral ions such as potassium, calcium, and magnesium ions from the soil into the root cells using active transport, a process that requires ATP. The accumulation of these solutes lowers the water potential inside the root, facilitating the continued movement of water into the root from the soil. Once absorbed, water and minerals move from the root hairs through the cortex and toward the vascular tissue. Water then enters the xylem vessels, which transport it upwards through the plant. The continuous absorption of water creates root pressure, a positive pressure that can push water into the base of the xylem, contributing to the upward movement of water, especially when transpiration is low, such as at night. Water moves through roots by two pathways, the Symplast and the apoplast. In the Symplast pathway, water enters the root cells and travels through the cytoplasm, moving from cell to cell via small connecting channels called plasmodesmata. This pathway is selective because water must cross the cell membrane, allowing the plant to control what enters. In contrast, the apoplast pathway allows water to move quickly through the cell walls and spaces between cells without entering the living parts of the cells. This route is non-selective and faster, but when water reaches the endodermis, this layer of cells in pink here, it hits a waxy waterproof barrier called the Casparian strip, which blocks the apoplast root and forces water into the Symplast so the plant can regulate what enters the vascular tissue. The Casparian strip is crucial for efficient water and nutrient transport in plants, as it helps to prevent the unregulated influx of water and minerals into the central vascular cylinder in the root. The strip also protects the plant from potentially harmful substances in the soil. After water and minerals reach the xylem through the Symplast and apoplast pathways in the root, they are ready to be transported upward through the plant. This upward movement plays a crucial role in the next phase of transport, translocation, the movement of sugars and other organic compounds through the phloem. Translocation is the process where organic compounds such as sugars, amino acids, and hormones move within a plant through phloem tissue. The pressure gradient is essential for this process and is part of the pressure flow hypothesis, the widely accepted explanation for how translocation works. A pressure gradient is defined as a difference in pressure between two connected regions, such as the source and the sink, which causes fluids to move from high pressure to low pressure. An example will be explained here, whereby sugars produced in the leaves from photosynthesis are transported to a sink such as the roots for storage. The process of translocation requires transpiration to create negative pressure, active transport, passive transports, and as mentioned earlier, a pressure gradient. Let's look at the process of using the numbers shown in this image of vascular tissue, xylem and phloem. Translocation moves sugars from the source, shown as a cell in a leaf, to a sink, shown as a root cell. Looking at 1, transpiration begins the process by pulling water upward through the xylem due to the evaporated water from the leaves. This creates a negative pressure, or low water potential, at the top of the plant, which draws water from areas of higher water potential in the root upwards through the xylem vessels. While this is happening in the xylem, another transport process occurs in the phloem, where source cells, typically leaves, shown here at 2, actively load sucrose into the phloem sieve tubes using energy. This accumulation of sucrose lowers the water potential inside the phloem at the source end. As a result, shown here at 3, water moves into the phloem from the adjacent xylem by osmosis. Since water flows from areas of high to low water potential, the influx of water creates a high internal pressure in the phloem at the source, which drives the mass flow, shown here at 4, of sugary sap towards areas of lower pressure, known as sinks. These are parts of the plants that use or store sugar, such as roots, fruits, or growing tissues. At the sink, shown at 5, sucrose is unloaded from the phloem, which raises the water potential in that region. Consequently, water moves out of the phloem back into the xylem, completing the cycle. This entire mechanism is driven by water potential gradients and pressure differences, efficiently moving both water and nutrients through the plant. As a summary, here you can pause the video to read through the 10 main ideas covered in this key concept video and check your understanding before moving on in your studies.